Hello, bachcho. Let's see the question. So the question says, which of the following is an example of direct gene transfer? So इन चारों ऑप्शन में से हमें आइडेंटिफाई करना है ये कौन सा टेक्निक डायरेक्ट जीन ट्रांसफर का एक एग्जाम्पल है. So the key concept in this question is direct gene transfer. So this is an easy question, and we need to first understand what is direct gene transfer. So direct gene transfer is the method in which DNA or the gene of our interest is transferred into the target cell directly. So in this method, we do not require a vector. So we can say that direct gene transfer is a vectorless method of gene transfer. So for recombinant DNA technology, we had to introduce the gene of our interest into a cloning vector, and that cloning vector transferred the gene of our interest into the target cell or the host cell. But in this method of gene transfer, we can directly transfer the DNA of our interest into the target cells. So there are several different types or different methods which can be used for direct gene transfer. So some of the methods of direct gene transfer are as follows. So the first one is microinjection. So microinjection is a physical mean of direct gene transfer where we can transfer the gene of our interest or the DNA using a micropipette into the nucleus of the host cell. So microinjection is a method in which we utilize micropipettes. So the gene of our interest can be transferred with the help of micropipettes, and these micropipettes can be injected into the nucleus of the host cell, and therefore we can directly transfer the DNA or the gene of our interest into the nucleus of the host cell. The second method of gene transfer is electroporation. So electroporation is a method in which we utilize high voltage for a short period of time, and this high voltage causes the formation of pores in the cell membrane of the host cell. So because of the formation of these pores in the host cell, that results in the entry of the DNA or the gene of our interest into the host cell. So with the help of high voltage power, the cell membrane of the host cell results in the formation of the pores. And due to the presence of the pores in the cell membrane, the DNA can easily enter into the host cell. That is, the gene of our interest can easily enter. So this is again a direct method of gene transfer where we can directly transfer the gene of our interest through the pores present in the cell membrane of the host cell. Now, the third method, which is a type of the direct gene transfer, is the particle gun method. So, in this method, around 1 to 2 micrometer gold or tungsten particles are first coated with DNA or the gene of our interest. Now, this uh, gold or tungsten particles which are coated with DNA is shot into the host cell with the help of a helium pressure gun. So this helium pressure gun is used to directly shoot the 1.2 millimeter gold or tungsten particles which are coated with the DNA into the host cell inside the cytoplasm of the host cell. So this is one of the most effective methods of direct gene transfer, because it is a sure shot method where the DNA will be transferred into the host cell. So these are the types of direct gene transfer with the help of physical means. Now, if we see the options given over here, then the first option says microinjection. So microinjection is a type of direct gene transfer, as we just studied. The second option says electroporation. Again, electroporation is also a type of direct gene transfer. And the third option is particle gun. So the particle gun bombardment method is also a direct gene transfer method. So we can see that all the three options are the correct option. Therefore, our final answer is option 4, that is all of the above. So the final answer for the question is option 4, that is all of the above. Hope you understood this well. Best of luck. Thank you.